I would like to first note again that we will be recording this section for posting to YouTube. So be aware that the recording is happening. If you speak or have a question, just know that that will be in the recording to be posted. I would also like to request that you keep your microphone muted if you're not speaking or having a question. However, this is meant to be a casual conversation type of lecture series. So if you have a question or a comment, feel free to unmute yourself at any time and simply say your question. At the same time, if you're maybe embarrassed or for any reason prefer not to speak out loud, you are welcome to put your question in the chat to me privately, to Sharer privately, or just to everybody, and we can make sure that that is asked. Besides that, I would like to just formally welcome Dr. Kiaei. A couple things I want to say about him. First of all, he's the owner and founder of RockGen Therapeutics, and I'm really looking forward to hearing his experiences about that. He's also associate professor at the University of Arkansas. He is the president and one of the original founders of our community, the Iranian neuroscientist community, so he has very, very unique experiences and things to speak to. So I'm very much looking forward to his presentation. And he will be talking to us about his academic and professional journey. So without further ado, please welcome Dr. Kiaei. And Dr. Kiaei, you're welcome to share your screen at any time.
Sure. Thank you so much. باید ببخشید که من انگلیسی حرف میزنم ولی میتونم این بحث ازشم فارسی هم بذارم چون اسلایدها انگلیسی هست بعد یه سری کلمات هم کم میارم به فارسی اگر به فارسی با من حرف بزنم برای همین شاید این مقداری فلوئنسی بیاد پایین. خیلی خوشحال هستم که این گروه دوباره به فعالیت خودش داره ادامه میده و دوباره میتونیم دوره هم جمع بشیم با هم صحبت بکنیم بعد مسائل کلابوریشن رو بتونیم بیاریم جلو با هم کلابوریت بکنیم و ساینسمون رو بتونیم ادوانس بکنیم. من امشب واقعا فکر کردم که افراد خیلی دیگه هستن میتونن بیان سخنرانی بکنن چون اچیومنتشون خیلی خیلی بیشتر از من هستش و به خاطر اینکه کسی نتونست بیاد من فعلا دارم جا خالی پر میکنم. برای همین این تاکی که دارم میدم یه چیزی همچین بلودای است. داستان زندگیمون داستان زندگی علمی رو براتون میگم و یه مقداری هم کارایی کردم حالت اسنپ شات داره میگم. بعد منظورم اینه که میخوام نشون بدم که اون عقیده اولیه که من داشتم تا حتی در زمان دبیرستان در ایران بودم که چرا میخواستم مثلا فلان کارو بکنم، بعد اون چجوری منو به اینجا رسون بدون که بدونم چجوری باید به اینجا برسم. و یه سری از افرادم در خانواده زندگی در اطراف به من اینسپیریشن دادن که بیام تو این خط تحصیلی و دانش و تحقیقات کار بکنم و هنوزم we are in the progress work in progress we have not done yet. خیلی زیاد مونده ولی اون چیزایی که تا حالا به دست آوردم فکر میکنم که کانتریبیوشنی بوده که من خودم ازش راضی هستم و فکر میکنم که مفید بوده برای فیلد تا حدودی و امیدوارم که برای شما هم جالب باشه. شاید بعضی جاهاش خیلی اسپشیالایز باشه ولی it's a type of you take as an example. اینو خواستم به عنوان مقدمه بگم. بعد خیلی اینفورمال میریم جلو صحبت میکنیم، سوال داشتین بفرمایین و با هم صحبت میکنیم. خب اجازه بدید من اسلایدمو شیر بکنم خدمتتون. اوکی میبینید اسلایدو؟
بله دکتر کیایی میبینیمش.
اوکی بذار من برم روی
فکر کنم دکتر کیایی همون دکمه اولی که داشتین درست بود. بله بله
اوکی
همون.
حالا خوبه؟ تمام.
بله درست شدن.
Okay, so in a Slido, I just made this slide just a couple of days ago to just give you a sort of global view of where I was to begin with in Iran and finish my high school. And then from there, I traveled to New Zealand. This was after I did my military service and was able to get my passport because I really wanted to go and further educate myself. And why did I want to do that? Because during the school, I was always wanted to become a doctor. And without knowing how, everybody asked me, what do you want to be when you grow up? And I was going to say, I want to become a doctor. And there were a lot of observation I had during my studies. For example, I'll tell you about that with more. My grandma was ill and I was going to be able to study medicine, so in a way I can help her. And many other family members I had sick. That was one of my motivation. So I left Iran in 1986 to go to New Zealand with not knowing any English and with not having much money either. And I landed in New Zealand with only $400 in my pocket. Imagine that going there, not knowing anyone, and how could you start with so little money and not even knowing the English? So cut the story short, I was able to get a job and learn English and then get accepted to university a couple of years later after I arrived and go into the path that I wanted to go from beginning. Then I went to University of Otago. I entered medical school and halfway through I decided to pursue a PhD. And I got my PhD and I just didn't want it to stop and go back to finish my medical school. So I decided to completely go to science and not wait any longer. Then at that time I got married and my wife said, she was in Iran, we came to New Zealand. She said, oh, I'm bored here. So let's go somewhere that is not as boring because New Zealand is a very small place. So then I was looking for a job and I got a job as a postdoc in New York. So we decided to come to New York. And here we flew over to New York in 1997. And there really, it's making the path for me from academic to now start up a company as a biotechnology company that I will tell you about that later. Then here, this is the website for my company, rockgenbio.com. If you have a moment, go and look and I'll be interested to know your opinion. So here, where I was in New York, and here the title of my talk, that a journey that in academics and from there go to biotech company in pursuit of drug development for the treatment of neurodegenerative diseases. And I'll tell you about that. Why did I go in this field? So from Dunedin, New Zealand, Dunedin is a city that Otago University is located here, a famous clock tower. It's very old and sort of big monument of university there. And so I came all the way to New York, and here this is the Mount Sinai Medical Center, Annenberg Building. Some of you may know about this. And I was here at the 20th floor. And there I got a job with the person who created first transgenic mouse. And I chose that lab because he was looking for someone to study ALS or Lou Gehrig disease or motor neuron disease. And I wanted to study ALS because I'll tell you a little bit later, my grandma here, in next slide, I will show you why I wanted to study ALS and the people behind the inspiration that gave me to go and study this field. So two grandmas that had a big influence on me and my great uncle. So my first grandma, who I told you, she was paralyzed from waist down. She had lower motor neuron disease. And my second grandma is my mom's mom. At a later age, she was suffered from Alzheimer's and dementia. And it was really sad when I went back to Iran and she could not recognize me. And that was another reason. Then the third person who influenced me, my great uncle, who was my principal in my middle school, and he got Parkinson's disease. And for all of them, there was no medicine to treat them. So these were the people who inspired me to go in this field. And then from Mount Sinai, I studied, my first project was looking at the role of copper in motor neuron disease. And working with pioneering transgenic technology, a scientist called John Gordon, who was the first person to microinject plasmid, linear plasmid, to the one-day old embryo and retransplant that to foster mothers and then generate transgenic mice. And I went there to learn the technology from him. And later on, you will see that I was able to utilize that technology and produce a model for a disease that we could study. And after 3 years, I moved to Cornell and worked with a renowned neurologist and neuroscientist, Flint Beal, who came from Harvard, headed the neuroscience and neurology department there. And it was a fabulous environment to work. And I worked there from 2003 to 2011. And climbed up the rank and became assistant professor there and managed to get an NIH grant. Then I'll tell you from there, I went to Arkansas because I was recruited there at the University of Arkansas for Medical Sciences to set up my lab there and continue my studies in ALS primarily, and then other diseases if I have additional resources. And this is the campus for University of Arkansas, the only medical school in the state. It's sort of equivalent of Harvard of USA. It's a very, very strong and high standard and high quality hospital, university, and research going on in this campus. So I mentioned about the influencers, and now let me just briefly mention what is ALS for people who are interested to learn about that and perhaps be introduced to this incurable disease, which is so far. So ALS is short for amyotrophic lateral sclerosis. And A means myo, no muscle. Myo means no muscle. A means no, and trophic nourishment. So that's where the amyotrophic means. And lateral sclerosis refers to involvement of lateral corticospinal tract neurons that they get stiff due to the nerve damage and their death and forming scars. So overall, ALS is a degeneration of somatic motor neurons that extending from upper motor cortical pyramidal neurons in the motor cortex to the lower motor neurons and brainstem and spinal cord. And I just want to give you a bit of a historical perspective in when ALS was discovered and what has been done in terms of clinical work and research. So in 1869, a French neurologist, Dr. Jean-Martin Charcot, first time described ALS as a pure amyotrophy. And it was for a long time, only neurologists could study the phenotypes and symptoms and clinical information of the disease and nothing else. So coming a long way, in 1993, a lab in Harvard, Bob Brown lab, they identified a mutation that associated with ALS, and that mutation called SOD1, superoxide dismutase 1, and was published in Science in 1993. And about a year later, this mouse model, the transgenic mice that overexpress mutant SOD1, was developed. And the same technology that John Gordon, my mentor, has pioneered in Mount Sinai. And this mouse, now you can see the hind limb is fully paralyzed and cannot even lift itself. So this is a full ALS symptom, recapitulate the disease phenotype very well. So if you look at that, about overall 300,000 papers published on ALS over 130 years or so. And out of that, over 100,000 of that papers published over 20 years from the time that this gene discovered and onwards. And in 1997, this is the landmark time that I arrived in Mount Sinai. And in 2008, there was first cellular model of motor neurons using induced pluripotent stem cells was developed and was discovered. And then I'll tell you more about this timeline down the line. So let me give you a little bit of anatomical perspective on where are the pathologies in ALS by looking at just the anatomy of a person that may get the disease. So this is the spinal cord, and this is the motor neurons coming here. These dark, large cells are motor neuron. And these are the cells that they die. And these are the cells coming here from ventral root connecting to our skeletal muscles. So when they die, then the muscle become disconnected and become paralyzed. The sensory neurons here, the dorsal ganglia, these neurons actually, they're spared and they don't degenerate. And we don't know the reason why these neurons degenerate and why those neurons don't. Perhaps the functional differences between the motor neurons and sensory neurons could have something to do with it, and then the high metabolism and continuous work in the contracting muscles and so forth. So they are under heavy burden of the task that they have. So anything that could make them very vulnerable. So this could be one of the reasons why these neurons are more vulnerable than the sensory neurons. So in the brain, this is the motor cortex, and where you have the premotor and then primary cortex. Here you have the neurons from here. They connect to brainstem and then come down to the spinal cord. So these are the area that you will have the pathology. And in patient, you'll have very few of these neurons left. And you need to have about 30% of them die before they actually get weak and show their symptoms. Now, how are we doing? Do we need to have a pause? Any question? No? Okay. So let me continue. Here, I like to give you a bit of a historical in terms of the number of genes discovered since the first one, SOD1 gene was discovered and the first mouse was made. It was for a long time, not much happening. And then later on, this alsin gene and many other genes start developing and getting identified to be associated with disease. And there are some interesting one like this TB43, stand for TAR DNA binding protein 43, that when this gene was discovered, they found a lot of pathology like aggregations of proteins in the tissues of patients that was studied, and they found a lot of patients have this not necessarily the mutation of it, but also the pathology of it. If they don't have the mutation, they have the pathology of it. I just remember to tell you that ALS, there are two types. There are familial ALS and there are sporadic ALS. Excuse me, I should have mentioned that earlier. And those 10%, 15% of the patient that they have the genetic inheritance of the disease, and these are the genes getting identified in those patients. So far, we don't have any gene identified in a sporadic ALS because there's no inheritance pattern. But there are potentially a combination of environment genes could be the etiology for the sporadic ALS patients. So these are the number of genes getting identified, and each of them have certain different functions. And in a way, it's very unrelated, but all of them give rise to motor neuron disease. So it's perplexing, and we don't really know how they are actually related that give rise to the same disease. And this gene here called C9orf72, which there is no protein being identified for this locus. And this happened to be a large portion of ALS patients, they have this mutation. And it's actually a type of expansion. So you have 4 Gs, or 4 guanine and 2 cytosine. They expanded in some patients up to 1,000 times. So it is very interesting, and it's very important because it's about 35% of familial ALS patients, they have this mutation. So then other genes discovered, like this profilin-1, that I put my lab focus on that. And we went and ahead and developed a model for that I'll tell you a bit later. By the way, this is the mouse model was made with the TB43 transgene, and this mouse is paralyzed and also is hunching back that with the help of Dr. Ismaili in my lab, we identified that these mice, not only they have neurological problem in their spinal cord, they also have problem with their GI gut. Their gut doesn't allow the food to be pushed down, so it got stuck. And then they have a tummy ache, and that's why they're hunching. And so we characterize that part of this model as well. So here, this is a picture of a neuron. So the whole idea is we only have one of these, and we would not be able to replace them if we lose them. What I mean by one of them, that is one of a kind. When this neuron is made and specialized and sort of have a particular function, it's very hard to replace it. And the stem cells and perhaps very rare, but they are not going to become another neurons to be replaced if this one got somehow injured and then died. So we have to look after this neuron and save this neuron as much as possible. And here a little bit of a diagrammatic representation of what is going on. This is sort of a little look like a third grader drawing of a neuron. And so in this particular cell, I'm showing a few sites of pathologies that, for example, if we have this porphyrin-1 mutation, what potentially could go wrong. And we could say the same thing with SOD1 or TD43 or C9 or 72. So when we have a mutation that the protein, this is supposed to be a protein produced, and it's malfunction or not function or aggregate, then that will lead to so many other pathways that could be disrupted. And for example, here mitochondria is affected with this porphyrin-1 mutation. We found that out when we studied the model that we created. And perhaps protein-to-protein interaction could be disrupted, actin dynamic could be interrupted, cytoskeletal, which is very important for the neurons, that some of them are very long, the axons could be about a meter, is affected. And then the nuclear malfunction, all the function in cytoplasm could be disrupted. And those neurons that we're supposed to look after, they are going to be dealing with these problems. And although they are very resilient, but they eventually will give up and then they die. And here, maybe I will not go through this. It's a little bit more details of where are the cytopathology due to what mutations. SOD1 causing a lot of oxidative stress, and TD43 and maybe optoneurons here, neurofilaments, and another cytoskeletal protein 2-4A. All of them are working in a certain area of this, and each of them by itself will kill the neuron. So I'll just sort of show that as, it's a mess going on inside a neuron, unfortunately. In a mitochondria, it has a double membrane, perhaps the SOD1 mutation will cause problem with calcium transport and with the ROS handling or creating more ROS, and the complexes aren't able to pass on the electron and convert ATP to ATP, the energy that the neuron really need to function. And all of that will cause lipid peroxidation, protein oxidation, and due to this type of proteins that they are mutated. So this is a sort of a mechanistic view of looking at what's going on in detail that I try to just sort of gloss over for now. So while I was going through this study from Mount Sinai, this is my first project in Mount Sinai that I looked at the role of copper, and if we want to know what was the role of copper in the cause of ALS. Now, you may ask, why did you want to study copper? And if I didn't have time to talk about the SOD1 enzyme, is a metalloenzyme that need copper in their active side to dismutate ROS, oxygen radicals. So having a copper in the active side and having the mutation in SOD1 will come and change the conformation of SOD1. And this copper is supposed to be shielded and does not get exposed to other electrons and other radicals, so it doesn't kick the oxidation, redox reaction in the cell. So we came up and we found out a mouse model, just a spontaneous model that has a mutation in the transport of the copper that reduces the copper in the body. So idea was, okay, if copper could be potential problem due to SOD1 mutation, not being shielded very well, if we reduce that copper, we'll be able to save those animals and they live longer. And this paper that we worked, it took us a while to finish it up and publish, collaboration of multiple people like Ashley Bush from Harvard and his lab in Australia helped us to measure the amount of copper in tissue. And here with John Gordon, who taught me how to make transgenic mice, and Flint Beal, my mentor in Cornell. So this work started from Mount Sinai, and then I finished it up in Cornell. So it took a while, and finally published it in Journal of Neuroscience. It's a paper that I'm very proud of because now there is a drug in the market by another colleague, Joe Beckman. Based on this study, they have identified a molecule called copper ATSM that somehow chelate the copper and regulate the copper. And this is a new drug now. It's getting to be tested in patients in Australia. So this work on the time that we really didn't have the future plan for it, but another investigator used that to make a drug, and hopefully that would help the patients. So here are the data that I'm not going through that, but I just want to mention to you that you do a work and publish it, and you don't know who will pick it up and what will they do with it. And I'm glad that Joe Beckman and his group, they picked it up, and they now coming up with a drug for ALS patients. And here it's a news about that as well, that copper therapy has an initial promise in ALS mice, and then that is going to be, I think the clinical trial is going on right now in Australia. Since I was interested, not only learn the mechanism of what is causing motor neuron to die, utilize those knowledge and identify perhaps drugs that already been FDA approved or other compounds that could potentially protect the neurons. And here in collaboration with a company called Celgene who developed a drug or made an analog of a drug, thalidomide, now called lenalidomide. We actually tested both of them. We started testing drugs in the SOD1 mouse model that I introduced early on. And here is a paper that we tested this drug and was able to delay the symptom at the time of onset. When you give it to the patient at the time of onset, this is the time that patient need the drug, because they go to the neurologist and get diagnosed, and then they needed a medicine. And that was our idea, giving to the mice at the onset of the disease and see if it's neuroprotective or not. And this drug was neuroprotective. Then I also was looking for other medicine as well to see which one of these potentially one day will go to a clinical trial. We learned about oxidative stress, as I mentioned, that has a big role in ALS, and we're looking at what are the molecules could potentially come and quell and slow down the stress and neutralize the proteins that they get oxidized or fix them. And this system called NRF2 antioxidant redox element system, we wanted to see if we can activate that known to induce about 250 genes that they are antioxidant, anti-inflammation, neuroprotective genes controlled by this system. And we found a collaborator who had a compound called CDDO etylamide, and there's another one, CDDO trifluoroetylamide. They modified it, and we tested in mice, and with a collaboration of multiple people. This was with Michael Spohn, who at Dartmouth, who was making these compounds. And then we tested in mice and found that this compound actually protects them very well. So hopefully one day there will be clinical trial. There is a company called Reata Pharmaceuticals. Now they are developing these drugs to be used, and hopefully one day they will test it in ALS patients. We also looked at other pathways and wanted to inhibit other pathways. One of them was inflammation, and this drug called Actos, peroxisome proliferator activated receptor gamma agonist, was tested in these ALS mice, and that drug also extended the survival of these animals, and we were successful finding a way that there are drugs that could delay the death of these mice. And later on in Arkansas, with the help of Dr. Smiley, who is here tonight, we tested another anti-inflammatory agent, which PPR gamma activator. Then that compound also protected these mice from ALS. So hopefully one day that compound could also be tested in ALS patients. So we are producing perhaps proof of principle. ALS is not incurable really any longer. Since we have demonstrated in a mouse model of a disease, then it could be tested in ALS patient as well. And I think I showed you this paper before, and we also been working on not only on ALS, on Parkinson as well, because as I told you, my great uncle had Parkinson. I wanted to see if one drug that worked from ALS could also work for Parkinson. And these CDO or triterpenoid, we tested in the mouse model of Parkinson in collaboration with Li Xuan, Yang, and other colleagues with Flint Beal at Cornell. Then we used MPTP, toxic mouse model of Parkinson, and we tested, and we found that coincidentally, this drug also protects the dopaminergic neurons that they died due to MPTP toxicity. So therefore, we found a common mechanism that NRF2 activation could protect the neurons in the ALS and in Parkinson, and extend this one further. My other colleagues tested that in Alzheimer mice while at Cornell, and we found that it actually protects them too. So it's very promising. Hopefully, these drugs could be tested. As you know, there's always a lag between when the science paper published and then by the time to go to clinic, sometimes could be 15, 20 years. Hopefully this goes faster. So I already mentioned that I went to Arkansas in 2011 and set up my lab in the University of Arkansas for Medical Sciences, and Dr. Smiley was my first postdoc there that we did some work I already mentioned. So this is sort of become really right in my blood now, that I sleep and dream about the science, how to protect the neuron in cases that could be hopefully helping the patient with the disease. I wanted to introduce you to a person that gave me a lot of inspiration while I was in Arkansas. This is Mr. May. He was the CEO of Siemens Bank, and he came up with ALS. And fortunately, his ALS is slow progressing. And this was a day that we went for a walk to defeat ALS. And a lot of Persian friends also came to help us. Here, my wife also coming supporting me, and my technician, Caroline, is there, and her father, and Hamid Habibi, Bishan Zandi, and Mehdi. Many other people came and helped us. So this is the moment that I will be satisfied with my work and say, look, if I couldn't help my grandma who passed away before, I come up with something, I think I'll be able to help this gentleman here who has ALS. So this is rewarding, and it gives me a satisfaction that, look, it is paying off. And if I didn't give a medicine to this patient here, but I gave him a lot of hope, at least at that time. And hopefully he will survive long enough and to be able to see the medicine that we are going to develop, and I will introduce that to you in a bit later. So going a little bit further in that historical time, I was telling you that when IPS drug-driven motor neuron was developed, and then other genes was identified in 2012, profilin-1 mutation was identified, science paper. Coincidentally, the person and the lab who identified SOD1 in 1993, Robert Brown or Bob Brown, came up with another mutation in profilin-1, identified this mutation, and they published that in Science. So then I thought this is an interesting molecule to make the mouse model. Now this is the time that I could use what I learned in Mount Sinai and go and make a transgenic mouse and produce a new model of ALS. You may say, oh, you already had this model. No, why you want another one? Because each mutation has its own pathogenic toxicity way that will cause a disease. So it's very important to get an idea how something else will cause the same disease. So we were lucky that we started this process and we made this mouse model. And this mouse model right now, as you can see, is dying from ALS. It's still alive though, but fully paralyzed. As you can see, it lost a lot of weight and cannot do anything. It cannot even write itself. So then, mouse is not the only model for ALS. There are other models produced too, fly, worms, zebrafish, and all of them are really needed and helping us. And because there are so many parts of the mechanism that we don't understand. And each of these models could tell us something different. So here, a paper that we reported, a profilin-1 mouse model of ALS. And as you can see, it was a large collaboration with a lot of people, a lot of labs involved. It took about 4 years to make this model, and I'm so proud of it. What it came out, it's really making a good contribution for ALS. And this is another postdoc that helped with this, Daniel Field. And then there was a news report on that we were interviewed, and I think it's got some popularity, and now it is in this mouse model has been given to Jackson's lab, and anybody can order these mice and study them. It's available to them. So let me show you the little bit of video of this transgenic mouse. This mouse here is a wild type. It doesn't have the gene, it's not sick. And this one has the mutant gene and it's sick. You can see the difference. So it's wobbling while walking in the bottom of the cage, while the other one is walking fine. And right now, this mouse, it has a full symptom of ALS. So maybe take about 20, 30 days from now and it will die. I think it's about 140 days of age right now, this mouse. As you know, a mouse will live about 2.5 years or so. And these animals prematurely die and fully because of the mutation that we put in this model. Here a bit of additional details. Normally, the way we want to see what are the phenotype these animals have, we lift up their tail and lift them up by their tail and look at the hind limb display. And you can see this hind limb are sort of pushed back, retracted toward their body. Typically, this is the wild type mouse, that when you lift them up, they open up their legs and look like they want to land as the airplane, open up its wheel. But here, these animals can, and then they, as they get older, this phenotype get progressed. Here, about 20 days later, you can see this phenotype get worsened, and this was the same mouse I showed you early on. And also, if you look at the leg muscle, this is the mutant leg, and this is the wild type. You can see the difference, how much atrophy in their skeletal muscle of the leg has been occurring in this. When you look at the weight of these mice, they start to lose weight and they diverge out from the wild type and non-transgenic mice. Here you can see they lose weight quite significantly and rapidly at around, and then they finish around 200 days or so. And the survival, also, this is the wild type animals living, as I said, about 2 years or so, but these animals begin to die around 150 days or so, and then they gradually die to around 270 days, and that's how long some of them live. Here a bit of a biochemistry. So we found the mutant profilin 1 we put in, it get aggregated. And then we came and separate the proteins aggregated versus non-aggregated. We call them soluble and insoluble, and run them on a gel. And here you can see in a non-transgenic mice, the soluble fraction, you have the band, which is the profilin band of protein. And here in the insoluble, this is I stand for insoluble, there's no band, there's nothing insoluble. And then in the wild type, where we put the wild type gene, no mutation, we get a lot more soluble because we are producing more of this wild type protein, but there's no insoluble. Where in the mutant, we get insoluble band. And that is the biochemical basis of the disease. Then it leads to the pathology, and that pathology will kill the neurons that I mentioned. And they also get a lot of ubiquitination. And why did they get ubiquitinated? Because the cell wanted to put them into the recycling and get them to be recycled with the ubiquitin system, and they get ubiquitinated. Ubiquitin itself is about 8 kilodalton protein that added to this and get them sort of labeled, say, cell or component of the cell to go and digest this protein away. But unfortunately, it can't, and that's why it accumulates. This is all profilin 1 ubiquitinated, found in the mutant. When you look at the insoluble fraction, in the soluble, there's nothing there. And also in the control mice, there are very little of them. This is just sort of a noise background. So coming back to the same model I told you, here as I showed you, profilin 1 mutation will lead to the aggregation and misfolded protein and insoluble, and then lead to multiple pathways that will cause disease and dysfunction of this neuron that cannot transmit neurotransmitter and connect to the muscle in this end, where the neuromuscular junction will be lost, and then the connection between the muscle and nerve will actually happen due to this. Since profilin 1 is important in actin polymerization, the cytoskeleton could be a major pathological path that these neurons will suffer from. Also want to show you what the aggregation of TDP-43 will do. that we found in the Profilin-1 mouse model, that there's nothing wrong with the TD43, but this Profilin-1 could be using as a seeding and aggregate the TD43 as well. And that was interesting for us, that now we have a pathological sort of similarities that is also found in human, not only due to Profilin-1 mutation, but due to the consequence of Profilin-1 mutation. These are the people helped us in the lab. Dr. Smiley is right here, and many others, technician students, and even a high school student here, and Caroline, and here, this is now Dr. Mina Nekui came from Iran, and I'll tell you about her work in a minute. And then other helpers in the lab, Daniel Phil, who helped out with the mouse model, and so many others. So these are all listed here, plus many other collaborators in other institutes, as well as the funding that was enabled us to do this type of work. So now this is my academic part, okay? Now, then how do I transition, or did I transition, or on the process of transitioning my academic work to go to a startup biotechnology company to speed up the process of drug discovery? So as you can see, all the drug testing, it took such a long time. From 1997 I started till 2017, we're still in the testing stage. So I said, we got to go faster and bring the drug to patients. So Mina was a PhD student in Shahid Beheshti University in Tehran. Each time I went back to Tehran to see my parents, went there and gave a seminar, and she became interested to come to my lab. So she wanted to do part of her PhD in my lab. And she was working under Dr. Qasimpour, and who Dr. Smiley was also working with. He was the head of the department. So Mina came, and before she came, she had a lot of problem with visa. That was the time with Trump got elected and her stuff got delayed. So in the meantime, she had more time to think about what she want to do and what she want to bring with her. Then she told me, oh, we are purifying components of saffron here. Shall I bring some to test? And here you all know about saffron, and such an amazing spice, had a lot of medicinal value, and Avicenna has described that in his book of Qanun of Medicine. And so here the review written on that, and looking at so many diseases could be benefited with this saffron. However, the number of tests has been done on saffron is very limited. And we know about anti-inflammation, antioxidation or effect of saffron, and many other, it helped mood. And also some of you may know consuming saffron could also make you be smiling and laughing a lot too. So it's a spice that makes people happy, and that could be a good reason behind it. So if you're healthy, you're happy, I guess, right? So she brought some component of this nice spice, and then we started to testing that whether this saffron compound could prevent oxidative stress, inflammation, and also maybe aggregation in this mouse model. There were some studies in Alzheimer's models that hinted that potentially the compound of saffron could be preventing formation of plaque. That is one of the major pathological reasons of Alzheimer's. So we did some testing, and we got interesting results. And then in 2017, I thought that this could be a good time to start up a company to speed up the process of drug discovery, starting from saffron compounds. So this company RockGen was born, and I call it RockGen because Little Rock, is a city that this University of Arkansas for Medical Science is located. So I name it, I put rock there and then put gen as part of genetic and RockGen. So I thought it's a sort of interesting name. And I had a lot of help. And I had a lot of people who helped me along. A famous chemist, Peter Crooks, who is the head of Department of Pharmaceutical Sciences in UAMS. He helped us in working at the analog synthesis of saffron compounds. And this is Nancy Rush, who is the department chair, that my also, department chair and mentor. Then Nancy Gray, who had the BioVenture, that had an incubator for companies to be formed and helped. And Kayatul Varghese, who helped with bioinformatics and crystallography and looking at the structure of proteins and how we can design drugs for it. And Bob Brown, who is a neurologist, as I told you, his lab discovered at least 2 genes associated with ALS. He is also a collaborator in the mouse model development. And this is Mohsen Sharifi, called Max Sharifi. He is a bioinformatician. He's a pharmacist from Moscow State University, and also he had a pharmacy degree from University of Kent in England. He is a great person to work with in designing new molecules. That he actually helped me to design a molecule for Parkinson's disease that I don't have time to talk about it now, but I'll just mention that. So now we have 2 projects going on in this company and trying to get funding from NIH. And we are submitting a small business grants called SBIR or STTR. And I just submitted one January 4th, just one for Parkinson's disease. And I have one for ALS is pending based on the saffron. So saffron compound analog that we made with Peter Crooks. So this is, as the historical time comes, it makes sense with me, and now we are here, and hopefully we have a lot more to add to this. So let's go to the next. So here is the little bit more details of saffron compound. This is called crocin-1. Very interesting molecule, has a sugar here and a sugar there, and then a linear carbon chain. And when it gets to the gut, this gets cleaved off, the sugars get cleaved off, and this part of it called crocin, it will go inside the cells and then to the blood and hopefully to the brain. And our analog is based on this compound. And here is a bit of a flow chart of what we do. So we synthesize, synthesize a compound based on the natural compound like this, and profile them and look at the drug profile and how could be a drug likeness of it. And then we will do the preclinical testing in the disease models that, like in my own mouse model or SOD1 or other available model, depending which one is more appropriate. And we are targeting these 3 diseases, ALS, PD, Alzheimer's, really in a way to help the patients who suffer the same way that my own grandmas and my own great uncle suffer from that disease and did not have cure. And then we will do the endpoint data analysis and validation. And then we'll go to phase 2 study, and then further down we do the hopefully phase 1 clinical study in collaboration with clinician. Here I'll give you a little bit of information of our Parkinson drugs. Many of you may know L-DOPA or levodopa is the drug that Parkinson get treated right now. And here, this is an enzyme called dopa decarboxylase, and L-dopa will come here and get converted to dopamine. And as you know, the main problem in Parkinson's disease is the lack of dopamine. And the cells that they produce dopamine in substantia nigra area, they die and they do not produce enough dopamine. So the idea is if you give them L-dopa, which is a prerequisite to make dopamine, then you will perhaps help the cell to produce dopamine. And it works. It works quite a bit, and it works very well, but it has a lot of problems and side effects, and not getting enough of this compound into the brain, because this enzyme is ubiquitous. It's found everywhere in the body. Therefore, we need to do something that this compound get delivered to the brain a bit more, and also help the neurons that produce dopamine, because they go under a huge burden of producing dopamine, heavy metabolic burden. And so with Max Sharifi, we came up with the idea of somehow chemically binding our compound to this L-dopa, and then delivering it to the neurons. So the neurons that they take L-dopa, they will have something that could help. And that compound is actually antioxidant. And that antioxidant could protect the neurons from oxidative stress, from inflammation, and give them L-dopa so they could produce dopamine. So we are going with two sort of situation strategy. One, continue using L-dopa as already been proved and working to patients that they make dopamine, and also protect those neurons, because we only have few of them left in the brain, and they are so precious. And so our project is hovering around this idea and producing. And here, this is the work with another PhD student at the Beheshti University, Shahir Saad, who is a bioinformatician, and helped us to design this, that it can somehow inhibit this enzyme in the periphery, but not in the central nervous system, where we need them to actually be working and producing dopamine. So that is the true idea of us doing work at RockGen. And I think I will end with that. And I want to thank you for your attention, for joining us this evening, and would be delighted to answer any question you have. And I hope you enjoyed it. Okay, I'll pass it on to you.
ممنون از سخنرانی عالیتون، لذت بردیم و امیدوارم که بقیه هم استفاده کرده باشن. اگر هر سوالی کسی داشته باشه به مدت خیلی کوتاهی میتونیم در خدمت باشیم، چون قراره که سوشیال ایونت هم داشته باشیم و که اون هم خودش قراره که 20 دقیقه یا 20 دقیقه باشه. اگه سوالی کسی داره.
آقا من سوالی ندارم ولی بسیار عالی بود. دست شما درد نکنه.
بله دکتر که خیلی ممنونیم ازتون. حالا من ببخشید برای اگه کس احد سوالی نداشتن برنامه‌ای که برنامه‌ای که ما داشتیم اینه که تو بریک روم بذاریم همتون رو و قسمت سوشال باشه در حقیقت و همین تا 1 دقیقه دیگه انجامش میدم
تا لیلی کارای سوشال در واقع ایونتمون رو انجام بده. برای بیننده هایی که از طریق کانال این ویدیو رو میبینن بگم که ماهای آینده هم ما باز ویدیوهای بیشتری خواهیم داشت و همینجا ما برنامه رو قطع میکنیم برای ریکوردینگ. ممنون.